Let us have a look on smooth muscle. The smooth muscle consists of spindle-shaped smooth muscle cells. They are usually connected by gap junctions in most organs. The nucleus is in the center. There's one single nucleus in the center. So this is longitudinal section. In a cross section, it would appear like that. So we got smooth muscle cells, also called leiomyocytes. There is a single nucleus in the center. This would be cross-section, and they are usually connected by gap junctions in most organs. If we look more closely, in more details, the surface is not smooth. It has various invaginations. The nucleus would be here. There are also organelles for synthesis of extracellular matrix, such as granular endoplasmic reticulum or the Golgi complex, because smooth muscle cells could be producers of extracellular matrix as well. And the contractile apparatus is attached to dense plaques and dense bodies. So we got actin, microfilaments, it's smooth muscle actin. And we got myosin. The sites of insertion on the membrane is called dense plaque. Insertion of actin inside the sarcoplasm goes to the dense bodies. And this would be actin and myosin filaments. And this pattern is spread across the cell in various directions, without any particular or apparent order in three-dimensional space. So there is no regular arrangement of these repeating units that would cause any cross striation. So this is the nucleus. This would be the granular endoplasmic reticulum and the Golgi complex. These invaginations are called caveolae, and the whole cell is inside or surrounded by the basal lamina. So if the smooth muscle cell with this distribution of contractile apparatus will contract, this would be the relaxed condition, okay, it could be shortened substantially and relatively even more than the striated muscle. So that will be the relaxed and the contracted smooth muscle cell. The smooth muscle cell contraction and the mechanisms behind that are partially different from the smooth muscle contraction mechanisms found in sarcomeric muscles. It will be discussed in more details in physiology classes, but just the basics for histology, for the purpose of histology classes, calcium again triggers the whole process. That's similar to the sarcomeric muscle. However, it doesn't bind to troponin. There is no troponin in smooth muscle. There is a calcium modulated protein called calmodulin instead, which becomes activated and it helps to activate the myosin light chain kinase into an active form, activated myosin kinase, and the myosin kinase will transfer phosphate to myosin, which thus, being phosphorylated, becomes active so the kinase phosphorylates the myosin, and this active myosin is able to bind to actin. So we got the contraction.